Thank you. So good morning, everyone. I'm going to start you off today and hopefully wake everyone up. So my name is Nichola Conlon, and I am the founder and the CEO of a company called Nuchido. And to begin with, I'm just going to give you a little background about this company, as it's fairly new. I founded it just under a year ago, and this was after a career in drug discovery, where I realized quite quickly that current methods are actually very inefficient. So I wanted to found a company that took different approaches to drug discovery that were far more sophisticated and less time-consuming and expensive. So I have to admit that this is actually my first time at a SENS conference, so I'm delighted that the speakers have asked me to come and tell you all about what we're doing at Nichido. So first of all, I'm going to discuss the use of network science in aging and discuss how aging is viewed from the perspective of a network scientist. I'm going to talk about the theoretical complexities that are involved in trying to mitigate the effects of aging and why this might often seem like it's going to be a really hard job to do. I'm going to talk about a concept called systems pharmacology, which is basically a combination of network analysis and chemical biology, and how this approach to drug discovery can be used to try and overcome these complexities that are associated with complex diseases. And finally, I'm going to show how we have had some success early on in trying to identify molecules that promote NAD enhancement in man. So first of all, I'm sure you're all very aware of the hallmarks of aging. I'm sure you all know everything about them. But I really like coming back to these, and I especially like this diagram because actually what it nicely shows is that these hallmarks are not standalone entities. They're actually all highly interconnected. So, for example, if you acquire DNA damage in a cell, this leads to other hallmarks. It leads to things such as mitochondrial dysfunction, accumulation of senescent cells. Then what you find is that these hallmarks then go on to influence other things. And eventually you get in a case where the original hallmark making more things worse. This is then going back and influencing the first thing, making that even worse. And what you find is that this means aging is driven by multiple vicious cycles. And as a network scientist, we like to refer to aging as what we call an inflected cascade disorder. And essentially what that means is that the system's running smoothly, one thing goes wrong, this has a cascade effect, it impacts a lot of other things, these then start going wrong, and eventually they feed back onto the first thing and make the whole system even worse. And because of this, aging demonstrates exponential progression. Aging is not linear, and unfortunately in reality this means that our next 5 years of our life is going to be markedly worse than the last 5 years of our life. So already at this sort of high level view, what you can see is that aging is very complex. But actually, this is a very simplified version because underneath each of these hallmarks of aging, we've got hundreds, even thousands of proteins that are all talking to each other in complex networks. So this tells us that if we want to do anything to try and slow or try and reverse the effects of aging, then it's going to be quite a difficult job. We're going to have to do multiple things to try and have any impact. If you try to just mitigate the effects of one of these hallmarks, you might have a small, short-lived effect, but actually everything else around it is still ongoing. So just trying to address one of these problems is never going to work. So based on what I've just told you, it seems like the outlook's fairly miserable. It looks like this is going to be a very hard job. I've told you that there are hundreds, even thousands of proteins that are all involved in this. I've told you that if we're going to have any impact, we're going to have to make multiple interventions, but how on earth do we calculate this? How on earth do we work out what the best combination of hallmarks is or interventions is to try and target? And this isn't just a theoretical problem. And I'm going to show you in this next slide a worked example. So here is a network diagram of all the proteins that are involved in mitochondrial permeability transition pore formation, which is involved in the pathogenesis of many neurodegenerative diseases. And what's the pointer for this? All the dots, they represent different proteins, and all the lines that are going between them, these represent all the crosstalk between these different proteins. So you can see that it's a huge mess. There are so many proteins even just involved in this one aspect of neurodegeneration. So how do we go about trying to find drugs that will solve this problem? Well, if I was to put my conventional drug discovery hat on from Big Pharma, I would say, right, what is it in this network, what is it in mitochondrial pore formation that's important? And I'd say, I know, let's look at VDAC1. That's important. I know what we'll do. We'll design a molecule to stick to it and we'll knock it out. Then it'll all be great, we'll all go home, job done. Well, in reality, what's going to happen if you knock this one thing out? Probably not much. You might get a small impact, but you're not taking any account of actually what's going on around it. Biology is really complex. It displays a lot of redundancy. If you're going to take this reductionist approach and you're just going to knock one thing out, then it's very unlikely that we're ever going to get very far. The other thing that conventional drug discovery tends to turn a blind eye to is the fact that drugs don't just stick to one thing. Drugs are promiscuous molecules, they stick to many things, and that tends to have a bit of a blind eye turn to it. You might be sticking a molecule to one thing, but actually what on earth is it doing to the rest of the network? How is it impacting the rest of the network in ways that you don't really want? And this is why a lot of drugs fail, because they've got unwanted side effects that just haven't been predicted. So, from a systems pharmacology point of view, how would we go about addressing this problem? Well, first of all, we wouldn't just look at one protein, we wouldn't just look at one target. We build a big network like this. We look at all the proteins that are involved, we look at all the crosstalk between them. And you might notice that some of these nodes, these proteins, have a larger node size than others. And what this size represents is a measure of betweenness. And betweenness essentially means the number of other proteins that a protein talks to that don't in fact talk to each other. So these proteins that have a high level of betweenness are actually weak points in a network. And that's because a lot of crosstalk is going via these proteins, and that there's not many other ways that this talk can occur. This is where there is a low level of redundancy. So you think that if you knock out some of these proteins instead, you're going to have a much higher impact on trying to break apart this network. And often these weak points in the whole process aren't the obvious ones. They're not the ones that Big Pharma come along and say, we'll stick a molecule to. They're not the ones that are most apparent to begin with. So if we can knock out these, we tend to see a much better impact on the network. And I've also mentioned that you can't just do one thing. So the same applies to this principle. We don't want to just knock out one of these proteins that shows high betweenness. We want to do a pattern of intervention. So we want to take out a combination. So we can analyze a network to work out which combination of these high betweenness proteins we can target to maximally disrupt and break apart the network. And this is where chemical biology comes in. So if we know the footprint that we want to target, we can identify molecules that have that exact footprint that will stick to the exact molecules that we want to and help reduce off-target effects. So does this actually work? Well, the sort of hit rate for the primary screen in conventional drug discovery is being published at about 0.1% of drugs that go through a primary screen will actually have potency and actually be active and do what you want them to do. With this approach, we have 20% success rate. In our primary screens, 20% of our molecules are active at nanomolar potency. So you can already see that such techniques in drug discovery are going to significantly reduce time and cost of this process. So if you want to look at drug discovery and molecular discovery for aging from a reductionist point of view, it's going to look like a very difficult job. If you're going to take narrow interventions, such as targeting one protein like VDAC1, and there are VDAC1 inhibitors in this world, then you're not going to get very far. It's going to be difficult. However, there's plenty of evidence now that conceptually simple interventions can actually yield substantial rejuvenation. For example, remove senescent cells. Conceptually, this seems quite simple. It does something good. So, can we do something? Can we translate these rejuvenation results? We're now in a sort of fortunate position where big networks are even easier to deal with computationally. And we also have lots of data now about chemical biology footprints and the footprints of drugs and what they stick to. So it seems like if we take more sophisticated and more sensible approaches to drug discovery, such as systems pharmacology, that we may actually be able to design molecular interventions that do mitigate the effects of aging. So I'm just going to cover some of the recent rejuvenation results. So here you can see we've got a variety of mice. Some of them are quite happy looking and some of them are not so great. And these have been rejuvenated by various methods, including you've got partial epigenetic reprogramming, selective senolysis, removing these senescent cells, pumping up NAD levels back to what they were when they were younger, and also young blood, old blood. And again, these sort of rejuvenation results on the outset look quite simple. Conceptually, you do one thing and it does something good. But again, if we want to translate these results into man, we can't get away from the fact that the underlying biology is far much more complicated. And if we want to get any sort of results like this in man, we are going to have to intervene on multiple levels. So that's what Nuchido has been trying to do. And as I mentioned, we're still a young company, and one of the things that we have targeted first is trying to restore youthful NAD titers in man. So I'm sure you all know the whole NAD story, but just in case, when you are young, you have very high levels of NAD, and then when you age, these levels decline exponentially. So you're looking at about 8.5 nanograms of NAD per microgram of protein in some human tissues when you're born, and then you get down to about 1 nanogram per microgram of protein at the age of 60. And experiments have shown that if you take an old mouse and you give it NAD precursors, you help maintain its youthfulness, as can be seen in this picture. So how have people gone about so far trying to translate this into man? Well, the predominant way has been by precursor supplementation. And I'm slightly skeptical of this because there's a few reasons. The first is that some of the actual precursors that have been used are very expensive when you scale them to the level that's needed to have the same effect in man. And also there's a well-known concept in biology that if you precursor into a system, the system will then establish an equilibrium again, which will result in a loss of efficacy. And there's some evidence already that has been published about in the data that there is a ceiling effect when you supplement with an NAD precursor, which basically means that when you're putting precursor into the system, you can't just keep putting more and more in because it's not equating to any further uplift in NAD. You also start to see lots of waste products which are suggestive of degradation of the precursor, and what this suggests is that simply there's oversaturation of the enzymes that are involved. So Personally, I view this as quite a narrow-minded approach. I view this as a reductionist approach. Doing this one thing by putting a one precursor into a system isn't really going to have much of an impact. So how did we go about trying to change this using systems pharmacology? Well, we did have some precursor supplementation. We had to give the system something to chew on, but we also have multiple interventions in our cocktail which are designed to impact all the networks that are involved in the enzymes that create NAD, the enzymes that chew up NAD, all the transporters of NAD. We then looked at how we could maximally upregulate this network, and we selected a combination of multiple targets and then used chemical biology to select molecules that had this specific footprint. We also paused for crosstalk by making sure that the molecules that we were using weren't having undesirable effects at other points in the network. And it turned out that the cocktail of molecules that was chosen were all generally recognized as safe, and we were able to go into testing straight into man. So this cocktail, I'll just refer to it as NCD201. So I'll just stress this is very preliminary data, and obviously you've got to start somewhere, and this is an n=1 experiment, so take that as it is, but I just wanted to bring something today to show you what we've done so far. So I'll just briefly go over the experimental trial that we did. So we took one male volunteer who was 57 years old. We measured the NAD titers in his blood in PBMCs. We spent a week before the intervention actually really measuring his blood quite a lot to find out what his natural circadian rhythms were throughout the day, because NAD fluctuates wildly throughout the day, and we wanted to make sure that we were actually going to be measuring an actual change rather than a change that was going to be happening anyway, say between the morning and the evening. So we established when this person's NAD baseline was always at a low point, so we knew that we were measuring a real increase. So we give them the intervention every day for a week, and this is the first set of results that we've got. As you can see, the data is variable, but especially on days 5 and 6, you can see that we were getting a significant uplift in NAD above base level. And just as a little side, on day 5, I know that the actual concentration of NAD on this day was 4.75 nanograms per microgram of protein. And just for a bit of fun, if you want to relate that back to the graph I showed you with the exponential decline in NAD, this would mean that on this day, this patient actually had an NAD biomarker age of 19, which for a 57-year-old isn't bad to tell them you've got their levels of NAD back to when they were 19. So on average, over the 7 days, we had a 381% uplift in NAD above base level. And just to compare this to a precursor supplementation method, namely that's been done by Elysium basis with nicotinamide riboside, you can see that, especially on days 5 and 6, we're doing significantly better than this. Now, we were aware that the data did seem a bit variable, and we thought this might be something to do with the components of the cocktail that we were using, so we decided to vary the cocktail slightly and try it again with a slightly different version. So we had a week's washout. We measured the NAD baselines again to make sure that they had gone completely back down to normal. And then we did the trial again with this modified version. And here you can see that the results are all less variable, and towards the end of the week, it's definitely ramping up the levels of NAD. We got an average of 272% uplift across this week. And again, just comparing that to precursor supplementation, you can definitely see that we're, especially towards the end of the week, getting a lot better than what you get with this precursor supplementation alone. And across both weeks, we had an average uplift of 331%. Now, this data is the same data, apart from now the baseline's different. So for this, we had another volunteer, which was a healthy 37-year-old male, so someone that was 20 years younger. Now the red line represents their baseline compared to our 57-year-old volunteer who was taking our supplement cocktail. So now if we can get anywhere near this line, that's a good thing, because these are the levels of NAD found in a 37-year-old male. And you can see that on most days we were close to it or even exceeding it. And we're quite pleased about this. And we got a statement that was from our university collaborator who measured these NAD experiments for us, which they said it shows that volunteer A's treatment takes him significantly higher than a seemingly 30-something healthy male. And when we told him that, he was obviously very pleased. So in summary of the data that I've just shown you, to our best knowledge, this is the first demonstration of the highest increase in NAD titer in man. But we have to stress that at the moment, this is in a man. But you've got to start somewhere. So now we're proceeding with further clinical testing. This includes a larger cohort of patients, and it also includes both males and females to see if there's any gender differences. We're also including extended exposure, as especially from the second cocktail, it definitely seems that increased exposure towards the end of the week is definitely leading to a ramp up in NAD. But it's fair enough increase in NAD, but actually what impact is going to have. So in the samples that we've got, we're also now analysing the other components, such as is there an increase in DNA repair and other downstream effects. And it is attractive to think that this systems pharmacology approach that we have taken has contributed to these beneficial effects. So for my last slide, I'm just going to finish on this graph. I made this graph some time ago, so it might be a little bit out of date, but last time I looked, I found a total of 56 interventions that had been demonstrated to increase lifespan in mice, and I've loosely grouped them into mechanism of action, which can be seen along the bottom. And the dark grey bars at the front are the number of interventions that I found, and the light grey bars at the back are the average percentage increase in lifespan that you got with these interventions. And I just wanted to make the point that in mice, if you can get a 20% uplift and increase in lifespan, and we can translate this to humans, then it seems like the longevity escape velocity theory does not seem that far-fetched if we can design molecules that could actually translate these advances into humans. So just to summarize, recent experimental results have shown that aging is definitely a process that can be stopped and reversed. We're in this weird sort of time when usually the public say to scientists, oh, can you not cure this? And scientists go, oh, no, you can't do that. And now we've got the opposite, where we're like scientists, and most biogerontologists now will agree that you can rejuvenate. And then you go to the public and they're like, oh, no, you can't do that. So we've got this bit of a weird situation where scientists are actually saying, yes, we can do something for a change. And lots of these systems and the targets that you need to look at are being verified now. But we need to bear in mind the complexity of aging is very complex. It's an inflected cascade disorder. So if we want to develop drugs or molecules or whatever you want to call them that are going to try and mitigate the effects of aging, then we're going to have to target multiple processes, have to look at all the hallmarks and the best pattern of interventions that will, we can use to possibly design drugs to mitigate it. So it seems that in order to do this, the systems pharmacology approach that I've just described could be a feasible way of designing molecular interventions. And just to sum up, to the best of our knowledge, I've just shown you some preliminary data that shows the largest NAD enhancement in man reported so far. Thank you.